Just like to introduce our next speaker, who is Dr. Andrew Isidoridy. Andrew earned his PhD in molecular biology from the University of Albany under the direction of Professor Robert Osuna, identifying and characterizing post-translational modifications of the transcription factor DksA in E. coli. He joined ProImmune in 2019 after completing his doctorate and currently works on providing innovative solutions for clients that deeply improve our understanding of both desired and unwanted immune responses. Andrew's going to be talking to us today in respects to evaluating immunogenicity risk of complex peptide products. So over to you, Andrew.
Okay, thank you. I appreciate that. Yeah, so as Jeremy had mentioned in my introduction, I'll be speaking a little bit today about some of the new regulatory pathways for evaluating immunogenicity risks for these complex peptide products. All right. So to just take a step back, I wanted to, the focus of my talk will be on generic peptides, but to kind of give a little bit more context as to what we're talking about here, some of the key aspects for understanding unwanted immunogenicity, the way we like to envision it is we like to see, we take a step back. We have our, you have your biologic, which would be processed and presented by an antigen presenting cell. This antigen presenting cell can either present your peptides from this biologic on an MHC pathway. Often this is an MHC class 2 pathway, or that biologic can elicit a cytokine storm and an innate immune activation directly. And so we do have a assay that I'll touch on called ProStorm that allows us to kind of evaluate that unwanted immune effect. But in addition to that, you have the adaptive response mediated through MHC class 2. And so we have a number of assays that also focus on this interaction between the CD4 T cells and the MHC class 2 pathway that's presenting this peptide antigen. And so we have the ProPresent assay, which is a antigen presentation maps binding assay that Jeremy had quickly mentioned. We have HLA binding assays, which use recombinant HLA alleles just to look for the peptide's ability to bind that HLA. And then we also offer T cell assays, T cell proliferation assays that look for the ability of that MHC peptide combination to drive T cell proliferation. And so, of course, all of that is happening upstream of other unwanted immunogenicity effects, such as cytokine production, B cell activation, and anti-drug antibody production. So when we're talking about synthetic generic peptides, there's a high demand. The generic peptide pathway gives you the ability to create low-cost, high-quality medicines. And so there's a high demand and a strong incentive to provide a pathway and allow regulatory access for this. But of course, safety and understanding potential immunogenicity is very important in that. And so this ANDA pathway provides exactly that. It's a regulatory approval pathway that relies on the kind of in vitro evidence that I just previously went over on my previous slide, and allows you to kind of build a picture as to what kind of potential risks your generic peptide might have. And so really the focus of this is on understanding how your generic peptide differs from the reference listed drug, specifically as it relates to impurity profiles. And so for any impurity that's found between 0.1 and 0.5%, the FDA is asking for that to be characterized, and for any impurities to be purified, and to actually evaluate whether or not those impurities are able to drive an unwanted adaptive response. And so just a quote from this, data should demonstrate for each new impurity that the impurity does not contain sequences that have an increased affinity for major histocompatibility complex, MHC, known as T cell epitopes, and that the proposed generic synthetic peptide does not alter the innate immune activity. So this is a field that's been very rapidly growing and changing in the past 12 to 15 months. And so as that's been changing, there's really two main immunogenicity focuses, and that's the adaptive and the innate side. And so when we're talking about the adaptive immunogenicity, really we're focused on peptide-related impurities. And when we're talking about innate immunogenicity, we're really focusing on process-related impurities. For the adaptive immunity, it has to do with those T cell epitopes that can be presented via the MHC to CD4 T cells. We offer our ProMap assay, which is an in vitro T cell proliferation assay. This utilizes healthy donor PBMCs and allows us to monitor for that proliferation directly. We also have a reveal competition assay. This utilizes a recombinant MHC binding model to look for the ability of your impurity or the ability of your peptide product to outcompete at a known given allele. On the innate side of things, really what we're looking at is process-related impurities and their ability to directly activate toll-like receptors. To evaluate this, we have a fresh whole blood assay we call ProStorm. It's a little bit different from our ProMap in the sense that we actually do our donor recruitment ahead of time, and the donors come in for a blood draw the same day that all of the analysis is done. And so within 2 to 3 hours of doing the blood draw, we're actually incubating with your drug product. And in this case, it would be the complete drug product and not components or purified impurities. So to focus in on the adaptive side, we utilize our ProImmune PBMC biobank. This is a series of cryopreserved PBMCs that we've generated from tissue from the National Health Service Blood and Transplant Service in the UK. And these PBMCs have been isolated, fully typed, and cryopreserved by ProImmune, and so they're very high quality. And we have a really nice sense of their HLA distribution as well. And so the way that this assay would work is we would take your peptides and your purified impurities and any other test articles that you wanted to include, and we would incubate them with these healthy donor PBMCs that have been loaded with the CFSE dye. And the CFSE dye is a flow cytometry dye that where in every kind of subsequent division, you get half as much dye as the original cell. And so as you can see here, cells in this quadrant B are kind of indicative of undivided cells, unproliferated. And as they proliferate, they move over here into this quadrant C. And so that's how we're measuring proliferation. It's a very sensitive readout. And so the key features for this assay, as it relates to using this for synthetic generic peptide regulation, is that really we need to evaluate a number of different lots that cover near release. So we want to focus on 3 lots that are near release and 3 lots that are near expiry or at the end of their shelf life. And that's for both the synthetic drug product as well as for the RLD, the reference listed drug. And then in addition to that, we would also want to include any purified impurities alongside. And so all these PBMCs, as I mentioned, come from fully HLA typed individuals. And so we can really do a good job of matching that to a general population, or if you had specific HLA types that you wanted to include in the analysis, we can accommodate that as well. And the sensitivity is demonstrated for this through a number of multiple concentrations. So we would typically do a peptide titration curve and include additional assay controls. And cell viability is also demonstrated with this assay, just as an additional assurance that any proliferation or any lack of proliferation isn't caused by cell death. So taking a kind of a bit of a different direction. So the other side of that equation is, of course, the innate immune response. And so when looking at the innate immune response, the FDA guidance recommends either using a PBMC, a whole blood, or a reporter cell line assay to determine risks. And these all come with certain pros and cons. Using PBMCs can potentially be easier, but of course, in the process of purifying PBMCs, you inevitably eliminate a subset of the population. Whole blood, we find, is a little bit more, we feel is a little bit more representative of the kind of in vivo condition of actually putting it in somebody's arm without having to do that. And then, of course, reporter cell lines are useful, but those typically focus each on a single TLR. And so you kind of have to be very careful of how you design an experiment that utilizes that. And so in addition to the whole blood, fresh whole blood, we utilize a panel of ERMIs. These are innate immune response modulating impurities. These are known impurities that target specific TLRs and elicit known cytokine responses. And then we would test your drug at the highest concentration we can that doesn't affect cell viability. And so these assays really need to demonstrate sufficient sensitivity, drug tolerance, specificity, precision, robustness, and accuracy. There needs to be a consistent sensitivity to low levels of a variety of innate immune response modulators. And I'll go over that here in a second. And then it also needs to be suitable in order to provide data that informs immunogenicity risk. It should be fit for purpose. So kind of focusing in a little bit on this panel of ERMIs that I was describing. We have a set panel right now that utilizes 5 ERMIs. These are endotoxin, FSL1, Clo75, zymosan, and miramil dipeptide. And all of these target different TLRs. And so that's kind of all listed here. And we do these in kind of a set series of concentrations that have been derived empirically and show really nice response curves. So just an example of how we would design such an experiment. So the ProStorm assay is a whole blood assay, as I mentioned. So we would actually recruit the donors for the assay to come in and ahead of time to come in for the same day that we're doing the assay analysis. We take the fresh whole blood, we incubate it with your test article. Typically for the pilot, we would do that over a series of high concentrations in order to try and determine the highest concentration that we can that's not affecting cell viability. And then we would allow those to incubate for 24 hours, at which point we would separate the plasma and we do an immunoassay to quantify a whole panel of different cytokines. And so that's listed out here. And we can also incorporate additional cytokines if for some reason you think that your peptide might be specifically eliciting a response from something else. And so for a pilot, typically we would approach these types of experiments as kind of a pilot and a main. The pilot is designed on a smaller scale, and it's really focused on kind of dose finding, finding the highest concentration of your generic peptide that's not affecting the cell viability. And it's also has a component comparing the ERMI panel alone, as well as the ERMI panel incubated alongside a high concentration of your generic peptide. And the point of that is to try and identify any potential suppressive effects or any other effects that your generic peptide might have on the ERMI readout as it stands. And then the ERMI readout also serves the function of establishing the sensitivity of the assay and really shows that we would be able to pick up any of these impurities even at very, very low concentrations. And so we do also include a cell viability component for this. And our kind of traditional assay controls are PBS and SEB, staph and enterotoxin B. The main study that we would do typically as a follow-on for this would come from, it expands usually to about a 30 donor study. And we would focus in on testing your complete set of batches. So again, this would be 3 lots that are near release, 3 lots that are near expiry for your generic peptide, as well as 3 lots that are near release and 3 lots near expiry for your reference drug. And so we would compare that alongside our standard panel of ERMIs. And this also helps, having the same panel of ERMIs in both the pilot and the main also helps to really establish consistency, intra-assay consistency, as well as really nailing home the assay sensitivity. And so again, we would be assessing cell viability here, and we have our kind of standard rest of the controls. So really, in summary, what I wanted to kind of make sure I drive home is that this is an area that's been rapidly evolving, that this is something that ProImmune is the experts, that we are experts in assessing immunogenicity. We really are on the forefront of this field. And we've been kind of working with generic peptides in this context since the beginning. And so just to really highlight how far things have changed, even since 2021, when that ANDA guidance was released, there's been a number of updates. And so to really hear it directly from the source, we would recommend checking out the FDA. There was a talk that we'll link out here to the FDA meeting from last September. Our assays in this regard feature the fastest turnaround time in the industry. We're very quick with this. Even the donor recruitment, we're very quick with. And so even something like ProStorm, we can get done within 6 to 8 weeks of contracting. We can provide the report. And so we work very closely with clients and to help support your regulatory success. And this has been demonstrated many times by successful outcomes of our customers. And with that, I just wanted to thank you for your time and just put up a little bit of contact information. We would definitely love to hear from you. And if you have any questions, feel free to reach out.
Great, thank you very much, Andrew.